Good afternoon, everyone, and welcome to this IZON Science webinar dedicated to Zeta potential measurement with TRPS technology. So, as you maybe already know me, my name is Dimitri Aubert. I'm the Global Sales and Marketing Director at IZON Science. And today I will co-host this event, collecting your questions during the presentation and submitting them to the speaker at the end. Questions that cannot be answered during the webinar will be dealt with later and by email. So this webinar is recorded and the link will be posted after the broadcast if you want to watch it again or if you want to share it with colleagues. And now I would like to introduce today's speaker, Dr. Mark Platt. He's a lecturer in the Department of Chemistry at Loughborough University. Mark's research interests include materials and microfluidics, aptamers, and biosensors. His pioneering use of the TRPS technology led to the publication of a range of novel pieces of work, some of which will be covered by today's presentation. And now it's over to Mark.
Thank you very much, Dimitri, for the introduction. I have my handler for the next hour sat next to me to make sure that I don't say anything silly and keep you all well entertained and educated throughout this process. Welcome to Loughborough. As you can see from the backdrop behind us, it's always nice and sunny. It's a pleasure to have you join us in the webinar. I should start by apologising for the delay in this by a couple of days, and that was due to just an illness on my behalf. And I'm glad that you've been able to reschedule and join us a couple of days later. So what do I hope to do in this session? I hope to give you a bit of an overview and the context of some of the measurements that we've been doing. Some of the potential of the technology is by no means designed to be an in-depth blow-by-blow account of Zeta potential theory and all the methods of analysis and comparison. I think that would go beyond the scope of what I have time for and what you probably wish to hear today. And I'm going to cover a little bit about the if you like, the work and the build-up to some of the methods and processes that are taking place. So what I hope to do is give you a bit of an introduction to Zeta potential, the what, the why, the uses, recap perhaps some basic theory of TRPS. I presume this audience is well educated in this, but I will just touch on it as a refresher so that you're aware of the key points that I'm about to go into. I'll go through the measuring Zeta potential with TRPS, and there are a few methods out there, and we'll go through them, at least introduce them to you. I'll give us some examples of Zeta potential measurements on TRPS, some of the kind of work that's being published, and I think at that point I'm not going to be able to cover the entire field, so I apologise if I miss your group or your favourite paper from this. This is really just some examples that we've been doing that I've seen recently from other groups. And then I thought I would just end on a couple of points for consideration, things where maybe the deviations and whether we should report as Zeta potential, is it applicable in the situation that we have. So let's start with a brief overview of the background of what is Zeta potential. And if we think about, we have some nanomaterials or particles that we wish to characterize, whether they are biological or inorganic. Some key parameters that we'd like to know about them would be their size, their polydispersity, their concentration, so how many we have in solution. And then another parameter is something to do with their surface chemistry. And those particles, if we place them into a solution, may have a charged surface, whether that is a charge inherent with the surface chemistry, whether it is a fact that something in solution has absorbed onto its surface, whether it be an ion or a surfactant, whether it's because that particle has dissolved or there's an anisotropic distribution of chemicals and functionalities on that surface that lead to an inherent charge appearing. But the placement of a charged particle into solution then changes the local environment. That is, the solution that immediately surrounds that particle is different from what would be in bulk, and what you would have found in the bulk of the solution prior to the addition of that particle. And what the the zeta potential then gives a parameter to is this electrokinetic potential between the surface of that particle and this local solution environment. And so the zeta potential isn't a specific number that talks about charge density or about the charge, but it is the potential between that charged surface and the local solution environment. By its nature, then, a negative zeta potential can be used to refer back to the type of charge on the surface. So a negative zeta potential would come from a negatively charged particle. And by the measure that I've just, if you like, described, it is a function of the material, this zeta potential. It's related to the charge density on the particle surface. It's related to the solution environment which you place that particle in. So the properties of the liquid, what's in that liquid, also affects the zeta potential. But it can be used as an indicator of the charge on the particle, and some people use it as a measure of the stability. So something that has a high zeta potential might be described to be stable, and it will stay in solution. That is, it won't aggregate or settle to the bottom of your container. And if when you come across quick searches for zeta potential, you'll very quickly come across a scale that I've got on the right-hand side of the slide here, where it says, highly charged particles are stable. And there's usually some sort of value that people will give that might say above this, a particle may be stable in solution and not susceptible to quick aggregation or flocculation. I don't think it's right to give a specific number for this because I think the zeta potential where particles are stable depends upon the size of the material, the density of the material. But as a general rule of thumb, the larger the zeta potential, the larger the charge and the more stable those particles can be. So let's try to put a little more detail and structure to what that local environment around those particles are. Here I've drawn a particle in the center, and it's surrounded with, and it's been placed into solution, I should say, with monovalent cations and anions of equal concentration. And I've drawn this particle here, it's been dropped into solution, and you can see around its surface I've drawn what would be cations. And the way in which I've drawn this diagram, then, what I've inferred and implied in this is that this is a negative particle. It has a negative surface charge. And when you drop a particle into solution, immediately on the surface, you'll get this strong interaction and absorption of cations onto its surface that are held in place. And then around this solution, you'll see this distribution of ions. And if you were to, if you like, shrink yourself down and walk around in this local area, you would bump into cations more frequently than you would do anions. That is, the concentration of cations around this negatively charged particle is higher than the bulk concentration, and the concentration of anions around this particle is much lower than the bulk concentration. So we can put some descriptors and terms to this. We have something that would be a surface charge. The surface charge has attracted these cations around its surface. These are held in place. At the outside of these cations that are held in place, we can use a term that's called the stern layer. Just beyond that, there's a boundary that I've drawn, which is the slipping plane, and I'll come back to that in a second. And then as you go a little further from the particle surface, you get this diffuse double layer. And this is an area that encompasses, if you like, this ion cloud, this cation cloud that surrounds this negatively charged particle. Now, the slipping plane in there is a descriptor that if I was to take this particle and quickly move it to another part of the solution, everything within the slipping plane would move with that particle. So every cation would move with the particle. And this diffuse double layer would stay put. So everything within that slipping plane moves with the particle. Then you move the particle to a second part, different location in that solution, and a new diffuse layer would establish itself around. So there are some terms there. We can now plot this electrokinetic potential versus distance from the particle, and you can see I've got and tried to match this up with the particle diagram. So on the surface we have a surface potential. On the outside of this stern layer we would have a stern potential. Just beyond that, at the point where the slipping plane is where we have our zeta potential. And then we carry on a distance from the particle, we get to this dashed line which represents the distance and the thickness of the diffuse double layer. Now we can never actually measure the surface potential directly, nor the stern potential. What we measure is the zeta potential. That's the value that we're able to measure easy, and that's what we'll go on to measure. And what we do is we relate that zeta potential measurement to the stern potential, the surface potential, and the surface charge via an equation, which I'll show you in a second. Now it's important to note that this diffuse layer, the thickness of this diffuse layer can be calculated. So this is the inverse Debye length, it's kappa to the -1. And simplified, it is related to, and you can see the parameter here, if I can just get up my little annotation. So this term here allows me, it says that this thickness by the inverse Debye length is related to the ionic strength. So the cloud that surrounds this particle, the size of this cloud will change if I increase the amount of ions in solution. So if I increase the number of ions in solution, the cloud, if you like, gets thinner. Okay, and that has a significant effect on the zeta potential, which we'll measure in a second. Okay, so we have this this relationship. We can kind of understand hopefully what the structure is around these particles. What is the zeta potential dependent upon? It's not a fixed value for a particle in a particular system. And it can change. It can change, and it's dependent upon the surface charge density of the material that you're analysing. And if we think about that, and we have a particle which has ionizable groups, carboxyl groups, amines, proteins, whatever it is, that can be protonated or deprotonated, then the charge density in those particles would change as a function of pH. And one of the important parameters that zeta potential is dependent upon then is the surface charge density, and as a consequence of that, that might be susceptible to pH changes. It's going to be dependent upon the solution environment and the type of ions that you add to solution. So some of these ions might absorb onto the particle surface, and that might change the charge density and the distribution of the double layer. And I've just shown you the equation that says that a double layer thickness is related to the ionic strength. And in fact, you can see beyond the double layer thickness, this electrokinetic potential drops to 0. So beyond the double layer, we go back to bulk solution and bulk environment. So the distribution of the electrokinetic potential within the double layer is distributed within the double layer. So if you change the double layer thickness, it will change the electrokinetic potential drop across that, and that can affect the zeta potential measurement that you measure. So the zeta value that you measure is a function of pH, and it's a function of ionic strength, and it's a function of the solution and liquid that you put it in, and the type of ions that are present. So when you give a zeta potential value, really sometimes you should really do is quote the pH, quote the solution, and quote the ionic strength that you've measured it in. And you know, for the same material, if you come along to make a measurement of a zeta potential on a different instrument under a different set of conditions, you will get a different value. So I said I would show you the equation. This isn't an equation that we have to linger on and we'll use as we go through the webinar, but this is the equation that says our zeta potential value here is related to the surface charge density and the double layer thickness. So if you can measure the zeta potential, you can then back calculate to what the surface charge density is, which is a useful relationship to have. Okay. So that hopefully gives us a quick intro to what zeta potential is, what are we referring to when we talk about charged particles, and when we're measuring it, what are we actually measuring, what is it susceptible to? How do we actually go about measuring it? So if we have our charged particle and into this solution, we stick 2 electrodes and we apply an electric field across it, that particle will interact with the electric field. And that interaction and the direction in which it moves and the speed at which it moves is kind of summarized in this electrokinetic effects. And there are a couple of different electrokinetic effects, but we only need to focus on 2 for the purpose of understanding today's talk. And the first one is electro osmosis. And that's the movement of a liquid relative to a stationary charged surface. And this is going to be more important when we start talking about our TRPS measurements. So I'll introduce it now and I'll come back to it later. As a way of an introduction then to measuring our zeta values, and to get them we need to focus on the second term, which is electrophoresis, and that is the movement of a particle relative to the surrounding liquid under an applied electric field. That is, if I have a negatively charged particle and I stick my electrodes in solution, it's going to move towards the electrode of the opposite charge. It's going to move towards the positive electrode, as I've drawn in that little cartoon in the bottom right-hand corner. Now, if we can measure the speed at which it moves, that's a useful parameter. So the particle's electrophoretic velocity, which is this VEP, and is related to the applied field strength, this electrophoretic properties, the electro-osmotic properties, and the diffusion constant of the material. And in the simplest way of setting up these experiments, we can make it so that we can assume that the electro-osmotic or diffusion are very small or insignificant contributors to this. So, and that simplifies that relationship so that we can now have this velocity. So if I can measure the speed at which the particles are moving, I can relate that back directly to the electrophoretic mobility. Okay, and that's a much simpler relationship because I've shown now that the zeta potential value is directly proportional to the electrophoretic mobility, and we can calculate that then from the use of the Henry equation. Here is that equation. So I have this term mu EL, which is our electrophoretic mobility. It's got a bunch of parameters in there. Let's just try and annotate this so we can go through it. Okay, so I've got a term here. This epsilon, this really just describes the solution parameters. It's the permittivity of solution. I've got a second term here which really just describes, again, the solution, which is the viscosity. So I've got a parameter here that says the velocity of the particle is related to the solution that you put it in. I've got, more importantly, this term here is our zeta potential. So if I can measure the velocity, it is related to the electrophoretic mobility, I have a term here that I can extract the zeta potential. Now I've also got here this FKA, which is, as you can see, I've underlined here, Henry's function. I've got this kappa term, which we've come across, which was described the thickness of the double layer. And the other term in here is A, and that is the diameter of the particle. And in most of the measurements that we have in the setup, we either assume or ensure that FKA follows the Smolkowski approximation. That is, the double layer thickness is much smaller than the particle's diameter. Okay. The alternative to that is where I have a double layer thickness, which is very large with respect to the particle diameter. And then that term here, FKA, is a different multiplier of this equation. Now, it's important that most of the assumptions that we have are where we have Ka much bigger to 1. That is, if I times the value via the radius of the particle, I get a value much bigger than 1. And in that setup, in measuring these in various different technologies, you assume that you've got a planar surface, you assume that you have a Ka much bigger than 1, and the alternative is if we have a very large diffuse double layer around our particles, the consequences is those double layers may overlap and interact with each other of neighbouring particles, which may perturb the measurement. And you also might get this effect, which is electrophoretic retardation. And so in the bottom diagram here, I've got this negative particle moving towards this positive electrode. In a very diffuse double layer, what you can also have is the cations in that double layer moving towards the negative charge electrode, which I show by this green arrow. And this is, if you like, is applying the brakes to the particle's speed. And as a consequence, then the velocity that you're measuring isn't a true value of the zeta potential. So in most of the situations, we ensure and assume that Ka is much bigger than 1 for simplicity. Okay, so techniques. I'm obviously going to talk about some sort of pole measurement, I guess, to measure zeta, seeing that I have a man sat next to me. There are other techniques. We'll all have come across these, so we've seen them referred to, sometimes asked to make an alternative measurement. They're all based upon exactly the same technology, or exactly the same concept, I should say. That is, if you take this electromagnetic light scattering, we're going to measure the speed at which the particle moves. We'll use that same relationship that I've just shown you there and take its speed and relate it back to a zeta value. Okay, and you know, in an ensemble technique like this, you get an average. So it doesn't matter how polar dispersed your system is in terms of size and charge, you'll just get an average value out the other side of it. And they're all, it's what I really want to say, and I don't wish to go into the pros and cons and the different technologies here, but it is just to underline, it's the same concept. You're measuring a velocity and you're relating that velocity back to a zeta value via the same equations, regardless of what that process is. So the theory, we've all come across this. I think I'm preaching to what is at least a converted audience or someone that's been at least read about this, but I think it's worth going through. So we have our setup, and at the top of it we have our pole, we have our membrane, I should say, and we have in the middle of this a pole. Okay, and our particles are going to go through it, and we'll use, and I'll show you a couple of different applications of a simple relationship to measure zeta in a second, but we should, I hope, know that the pulse frequency, that is the number of particles that goes through the pore per unit time, can be related to concentration. There are a couple of other terms in that equation, which I'll show you in a second. So you can get things like concentration out there. You can get the size of the pulse from the size of the particle, I should say, the particle volume from the delta IP. And the duration then, which is the thickness of this pulse, gives me an indication of the speed at which the particle is going through. So something that's going through really slowly and being dragging something along with it would have a very wide duration. Something that's racing through there would have a very narrow pulse, and we would say that was at a higher particle velocity. So this pulse width is a measure of how quickly the particle is going through the pore, and we've just introduced if you can measure velocity, you can work out the zeta potential. Now the leap then from measuring how quick something goes through a pore via the duration and converting that into a zeta potential, then hopefully it's not going to be too much of a jump, and actually would seem quite logical for us. In this setup though, and this is a slide that you will have seen or will be about to come across if you're about to embark on the training program through IZON, we can't simply just assume, as we did in the introduction, that it's only electrophoretic mobility that we have to worry about, and that other parameters are not going to affect the particle speed. So we have our particle and it's moving through the pore. We've got the one that we're really, if you like, interested in terms of electrophoretic mobility, because this is the thing that we can use Henry's equation and directly relate the speed back to our zeta potential. But there are other contributing factors to this. We have this natural fluid flow from the head pressure, where we put our sample at the top. And the fluid velocity is moving the particle from top to bottom, and if I change the amount of sample I put into the top, then that velocity will change. We also have electro osmosis, and this is the thing that I said we can, in the simplest setup, ignore or assume it will be a very small parameter. In the pore situation that we have, it's a charged pore, it has a slightly negative charge at a neutral pH, and therefore then we will have electro osmosis, and the electro osmosis is in the way that this diagram is set up with the orientation of the applied voltage bias, would have electroosmosis moving against the particle, if you like, trying to slow down the electrophoretic speed. So in deriving and coming to a relationship that says we can measure particle speed and relate that back to zeta potential, we have to have some knowledge or understanding of the fluid and the electroosmotic contributions to that to get a true accurate value of the zeta as it comes through. So just as I've shown you how the structure of a particle has a stern layer, it has a diffuse double layer, a charged pore would have a similar structure to it. And I think it's just important that at least we're aware of it. So we would have a charged pore, it would have a stern layer, it would have a slipping plane, it would have a diffuse double layer. And this is the nature and where our electroosmotic flow arises from. So we just need to be aware of it. I'm going to come back to the effects of this and perhaps how you would change it or what happens if this changes during the course of your experiment later on. But I think I just wanted to introduce this now. So in going through the literature with TRPS users, you'll see there's a whole bunch of people that have been working on zeta potential measurements for a while. They all have different ways to get to zeta measurement, and some really nice examples out there. This is one that come from Jeff and collaborators, and this is this equation that I introduced at the start, and now I'm going to give you the full relationship that says J, which is my pulse frequency. And I'll just get my draw and figure up. So here's our term that I introduced before, which I said was J, the pulse frequency, number of pulses per unit time. It's related to concentration, although I've got in the paper that Jeff's got, he's rearranged this and brought concentration up over here. And then we have a terms that says that the pulse frequency is related to the zeta potential of the particle. I've got the zeta potential of the pore, I've got an electric field, and then here I've got this factor that says the pressure, okay, if you like, the fluidic velocity. So a contribution to the fluidic velocity. Okay, so here is my contribution, if you like, from electroosmotic flow from the pore, and here is my contribution from QP over A is the contribution to the fluid flow. So in our experimental setup, we would usually add the particles at the top, so I'm going to start with the, down here in the bottom left-hand corner. Typically, the particles in an experiment would flow from the upper fluid cell to the lower fluid cell, and you would see your pulses. And indeed, in the graph that I've got here, I've got applied pressure along the x-axis, and it starts at 0, and I've got counts on the y-axis. So with 0 applied pressure, you've got the particles going from the top to bottom, and you have a particle count rate. And you know from the variable pressure module that you can increase, if you like, the applied pressure in P1 and you increase the particle count. What you can also do is, if you like, decrease the pressure in P1 by, if you like, applying a vacuum to it. And if you do that and you change the ratio so that P2 is bigger than P1, so I'm now moved over into a region where, over on the right-hand side of this, where P2 is bigger than P1, then particles would, if you like, move from the lower fluid cell into the upper fluid cell. So in the experimental setup that Jeff and co-workers came up with, they had particles in the upper fluid cell and they had particles in the lower fluid cell. And they went through a system where they varied the pressure, and as you vary the pressure, you get to a point where instead of particles flowing from the upper fluid cell to the lower fluid cell, you get to this region over here where you are now actually transferring particles from the lower fluid cell to the upper fluid cell. And going from that transition, you can see, if you like this, if you're a nice S-shaped curve, where you see an increase in particle rate, you see something leveling off, and then you see an increase in particle count rate. And this relationship is that if you can change the pressure, you can work out what pressure, if you like, stops the particle flow, particles going through the pore. So in going through this, you're setting J, our particle count rate, to 0. And in the paper, they can calculate the electric field. They've measured the variable pressure. And then this is a simple rearrangement of this equation where you plug in the values, you can work out the zeta potential of the particle going through this. And they can work out and go through and show the zeta potential of the particles that they've got. They compare it to a comparable technique, which is usually asked for, and I will come back to by referees in a second, and then go through and use those equations to calculate things like surface charge density. So they get, if you like, an average zeta potential of the system that they have, of the particles that they have in solution. And this was in 2012, so I guess Rob and Ben and Jeff have been working on different iterations and tried different things out. A couple of years later, Darby, Will, Rob, and others in Matt's group have come up with a slightly different variation on that. And they've changed it slightly so that instead of getting an average zeta potential for the particle distribution you're looking at, you now start to get individual, this particle by particle measurement, where you get individual zeta potentials for individual particles. And I'll go through the concepts here, and I've taken some of the data from this paper because I think it shows some nice if you like, effects of the system that we're working on. If you increase, if we have particles in the upper fluid cell and you measure their velocity as they go through, you can imagine you could change the, if you like, the applied pressure, you could have changed the applied voltage, and that might have an effect on the signal, it might have an effect on the duration. And what I'm showing, I guess, in the data, just to underline, and I think is an interesting point, is that if you apply the pressure, get my annotation, if you increase the applied pressure, what they've got in this paper are two particles, so I'm going to circular them here. They would have what they termed as a high charge and a low charge particle. And at low pressures, you can see that the high charge and the low charge particle have very clear differences in their particle velocities. And as you increase the applied pressure, those velocities converge. So you can dominate the flow, you can dominate the speed of the particle by the pressure and eliminate the effects of the electrophoretic speed. So it's something to consider when you're optimizing your parameters for your measurements, to make sure that you haven't had to apply too big a pressure from the system. What they've also got is a nice example where they've got now applied voltage. So I can increase the applied voltage and I can measure the pulse width again. And again, they've got the same labeling system. So this is a high charge particle and a low charge particle. And if you imagine, as you increase the applied voltage with the high charge particle, you see a pulse duration go up. And you see the opposite effect for a low charge particle. So as you increase the voltage, the low charge particle speed changes, and the reason being for this is that instead of increasing the speed as the high charge particle would, you're actually seeing electroosmotic effects start to dominate. So as you change the parameters in the system, you can see both the pressure and the electroosmotic effects of the pore start to take over in different situations. And it's an important thing to know, because you have to come back and calculate them, or at least account for them later on. So in the paper that in this one, if you like, the advancement was a measurement of an individual nanoparticle, and they go through and show how you can actually do a particle by particle measurement. And what they're going to do is use the pulse shape. So if we have our pulse, it comes up to if you like, a maximum, where I'm drawing here. This is our maximum increase in resistance, and this represents where the particle sits at the pore mouth. And then as this current decays back to the baseline, and the speed at which it decays back to the baseline gives you an indication of how fast the particle is moving through the pore. Now that speed, if you like, the current at any point in time can be calculated by the equation that they give, which is here. And if you please, I'm not going to go through it in complete detail, but it's worth pointing out that you can calculate the current you would expect to see at any point in time, and the current you would see is proportional to the length of the pore diameters, and importantly, there's a parameter here, D. Okay, and D can be broken down into its individual components, whether they are electrophoretic, electroosmotic, or pressure difference across the system. And what they have done in a really nice piece of work is where they calculate the individual contributing factors that would affect a particle as it goes through, whether it be osmotic, phoretic, and fluidic flow. And then you get the average, if you like, the sum total of those effects on the particle's velocity. So in knowing the electroosmotic flow through the pore and knowing the fluidic flow, if you then take a particle at an unknown electrophoretic velocity, you can then calculate back the Zeta potential of the particle as it goes through. And indeed, in that paper, they show the examples of how they've gone through to do this for a particle by particle measurement. And then a couple of years later, we had the fortune to work with Rob on an iteration of this method. And I should say that I'm going to try and do the best I can to talk you through this, and full credit to and acknowledgement to Rob, who really helped develop this. And this is very similar to the process that you would see IZON advise you to do. It's a slightly different iteration of that, where we can do particle by particle charge measurements, and I'll show you the example of it on DNA modified particles. This is a slightly different method. I argue it's a slightly simpler method, and the idea is that instead of having to calculate each of those individual contributions for the particle setup that you have, where you have to know the pore dimensions, and you contribute and you calculate the fluidic flow and you calculate the electroosmotic flow, you can use a calibrant, and you can measure the velocity of a calibration bead of a known zeta potential, and you can then relate unknown particles which you're interested back to this calibrant and calculate their zeta potentials. So it's going to rely on the same concept that we showed before, where we're going to look at the pulse. So I've got my diagram on the left-hand side here. I've got time versus resistance. This is my pulse that I've tried to draw. You have a pulse shape, and you're going to measure the, if you like, the width for the pulse at different points in time as you go across. And as you go across, as each one of these, if you like, different time zones represents the particle moving through a pore. And instead of taking an individual measurement where you might just say, well, let's just measure the pulse at half height or a certain time frame, the idea is that you would measure the pulse duration at all of these individual time points along the pulse, and you would take an average. And you would take an average velocity of this, and it's 1 over this average, which gives me my particle velocity. So the idea is in not doing an individual measurement, and instead of taking multiple measurements across that pulse, you get a better average, and it allows you to be a bit more reproducible in the measurements. So I plot the durations, I measure the pulse widths at different locations, and 1 over this average velocity gives me my particle velocity. And it's a way of determining how fast the particle is going through the pore. So the concept is very simple. If I have my sample and I measure its velocity, it's going to be related to the calibration's velocity by this equation. Let me get my annotation tool back up again. Okay, so this relationship between my sample zeta potential and my sample calibration, if you like, my calibration zeta potential is simply can be worked out by a relationship by saying, if my velocity of my sample is the same as the velocity of my calibration particle under the same set of conditions, then the zeta potentials must be equivalent. Now, in the setup that we have with the pore, we don't just have electrophoretic abilities, we have electro osmotic and fluid flows. So if we change any of the parameters and allow for any adaptations, we're going to need a bit more of a complex formula. So the sample, the velocity, if you like, of the particles it goes through in reality is contribution of the particle's velocity from its zeta potential, but a combination as well of the pore's zeta potential. So you're always going to get this contributing factor here because it's a much more complex system. We have the pore wall, and the pore wall is charged. So to account for this and build in a bit more flexibility, we're going to calibrate the pore by making a measurement of the particle's velocity at different voltages. Okay, and this allows us to then come back and make a measurement of our sample under different conditions under a fully calibrated pore. So I changed the voltage and I measured the velocity 1 over t for my calibration beat as it goes through. And in generating our calibration plot here, what we've been able to do is have a relationship that says, well, the electrokinetic velocity of the particle is proportional to the gradient of the graph that we've generated, and the convection velocity, that is the fluid flow from the pressure head from the top to the bottom cell, is now going to be the intercept on the y-axis. So I take those two equations at the top, and it's a rearrangement, and this is perhaps an equation that you don't necessarily need to put to memory. The software and the formula will produce these numbers anyway, but it will tell you at least where they come from. But it says that the sample zeta potential is equal to the velocity of the sample that I've measured, and I'm just going to get my annotation tool. So here's my zeta potential of the sample. I've got a measured velocity of a sample. The zeta potential of the sample that I'm interested in can be found out by measuring the velocity of the sample, subtract the if you like, the contribution for the convective velocity divided by the velocity of the particle, the electrokinetic velocity, times by the net zeta potential of the calibrant plus this contributing factor from the pore wall. So every time that we do a particle measurement, and this is the same for if you take an average of all the particles or an individual particle, this zeta potential of the sample that you're looking at can be derived from measuring its velocity and a rearrangement of this calibration plot. So you take away the the intercept divided by the gradient and times by the calibration zeta potential. And then you can account for the changes in the system as you go through and make the measurements of your sample. So this is as close to the technique and mechanism that IZON will, if you like, send out when they guide you through a zeta potential measurement. This is if you like, the equation which is being crunched in those spreadsheets. So I'll show you an example of that, and this is some work that Rob did. It was published in that Langley, where he's gone through and used that equation to measure the zeta potential of particles. You get these nice particle by particle measurements where you get distributions of zeta potentials versus particle size, and it will now pick up the individual particle sizes and their zeta potential measurements, and you get these nice distribution plots for these. I'll also show you a nice example, and I'll put up a few. Just as I said, these are ones that I've just have come across because of the work that we're doing in the lab, or I've seen recently in readings. It's some nice examples where people have compared the TRPS zeta measurements with alternative techniques, and I think as we go on and publish, it's worth knowing what people are doing in this area. So this is a piece of work that's come out of the Manelli group in the UK, where they've gone on to compare the size and zeta potential measurements of particles in serum using TRPS and ELS measurements. And you can see the comparison data in the table within the paper. I'll come back to the example where we working with Rob, where we demonstrated that the application of that equation that I just went through, and give an example where we're taking some particles and we're simply going to add to that particle a strand of DNA, and we're going to functionalize that particle with a length of DNA on the surface. And if we think about the zeta potential measurement is a function of charge density, so if I add more DNA to a particle surface, I increase the charge density around it. Each one of those phosphate groups on the DNA has a negative charge. You would expect to see a change in the zeta potential, and indeed, if you follow concentration of capture probe, and just so that we're all on the same terminology, I've got my CP concentration, which is my capture probe, and my capture probe is this strand here. So as you increase the number of strands on the surface, you would expect to see a change in mean zeta potential. We've plotted mean zeta potential, but you can also show the distributions of these particles as we come through. I quite like looking at distributions of particles. It gives you an idea of the distribution of ligands. Not all particles will have the same number of ligands on there, and it's nice to get a feel for what that distribution is. Okay. So as well as, you have a particle, you functionalize it with material, and one of the nice things about zeta potential numbers is it allows you to follow a reaction. You're changing the properties of the material, you can follow that reaction taking place, whether it be more DNA on a particle surface or you change the length of the DNA on a particle surface. And this is just another one of those examples from the paper where we have our mean zeta potential as a function of the number of bases, if you like, the length of the DNA that goes on the particle. You get a nice shift in the mean zeta potential, but you can also see a nice distribution change from the 10 rho to 50 rho. So, it's not simply just an increase in or a change in zeta potential, but you're changing the distribution, the width of the distribution. Ligands of different lengths will have different packing efficiencies on the surface, and having the mean value next to a distribution value is a bit of use, I think, to go through this. I'll show you an example where we really have to start thinking about what does the zeta value mean, what is it telling you, what can I get from it. The value that I just shown you in red was what we would call 210 nanomole of DNA we've added to the solution. That is, we've put in enough DNA to saturate all the binding sites on the particles. If you only had a fraction of that, so 75 nanomoles, so I'm no longer now saturated all the binding sites, what happens to the zeta potential that you measure? Do you see similar trends? What happens to the distributions? And you can see that you might get an increase or a change in zeta potential, but the distributions also change quite drastically as you go through it all. And I think one of the nice things was the ability to resolve those differences and plot those distributions as you're doing this technique. Now, I will show you this, and this is where we start to think of what does the zeta potential really tell you? If you take a value out of context or without some understanding of where it's come from, what can you get from it? What do you really want to get from a zeta potential value? This is the kind of experiment that we've been doing, where we take a particle and on the surface we put our capture probe. So this is the same example what I've just shown you. And then to that capture probe, I'm going to do hybridize some complementary pieces of DNA. So in the first example, which I named middle, I've got a strand of DNA that just hybridizes to the middle of the strand of the DNA. And an alternative experiment and variant on this is where I take the same length of DNA, but it's complementary to just the end of the complementary strand. Okay, so if we think about how our zeta potential is a function of surface charge density, what I've just told you there is I've taken the same amount of DNA, okay, the same length, middle or end, and I've added it to my particle surface. In theory, I've increased the surface charge density the same, okay? They should be the same lengths, the same amount of DNA. And if you were to step into the lab and then make a zeta potential measurement of these particles, are the zeta potential of these particles, are they the same? Would you expect the middle to be higher than the end, or the end higher than the middle? I just realized these are actually the same statements. So is it going to be the same? Is it going to be the end or the middle? And it's not immediately obvious, or it certainly wasn't immediately obvious to us when we started to go through doing these experiments. I'll let us think about it for just a couple more seconds, and I'll throw up the data for this. Okay, so here's that experiment. I've got the mid and end, okay? And the red, green, and blue are different hybridization times. I'll come to this. The green one is a 16-hour hybridization. So I've left those two things mixing together, where they've definitely picked up as much DNA as they can. And you can see, even though I've got comparable DNA lengths, comparable amounts of DNA on the surface, they give a different electrophoretic velocity through the pore. And that's interpreted then as a different zeta potential. Okay, and is it just related to the charge density? Is it just related to the solution? It's not, it's related to where that slipping plane is with respect to the surface, where the zeta potential value comes. So these are given different structures. The structure in that double layer, the structure of the DNA surrounding the particle is different. And we know it's different because we can measure different velocities, and they are recorded and transferred to different zeta potential values. Now, on their own, isolated, if I just gave you the zeta potential value of the end experiment, that wouldn't really give you too much information, I don't think, without the comparison and, if you like, the control experiment alongside it. So when you think about whether you want a zeta potential value, what does zeta potential value matters, you have to kind of really think about what are you implying? What am I assuming about the particle that I'm measuring? What am I assuming about the surface and the local environment around that particle and how it's changed? So the green plots here are after 16 hours. I've hybridized the particles and the target DNA for 16 hours, and you can see the full range of experiments there. So I start off, and get my annotation back up. I start off with my capture probe, and to it I add a cDNA, a fully complementary strand of DNA. I have this mid-T, which I've just explained is the middle binding section. I have an end-T, which I've just said is the end binding section of DNA. And we have something called over-T, which is a piece of DNA which hybridizes to the strand, but really has a big, long, overhanging section of DNA as well. And all of those green bars then represent a 16-hour hype. So you mix the two things together for 16 hours, and you can see these are replicate measurements. You can see the error bars of the change in zeta potential. So this is the change in mean zeta potential from the capture probe, which is essentially our blank. And you can see the largest shift for this overhanging T. You see different levels of shift in the different structures of DNA. And as well as being able to put a zeta value onto this, what was nice when we were doing the experiments was that you could resolve differences. So this is a technique where not only can you see a change in mean zeta with the distributions, but very small changes in structure can be resolved in terms of the experiment. The red bars on here, just to talk you through for all of the data, the red bars is that same experiment done in 30 minutes. And I think if you look at the error bars after 30 minutes, it'd be very difficult for me to be able to defend that these things are distinguishable and that I can isolate and detect these different structures after 30 minutes. But you can tune if you like, the assay conditions to what would be the blue bars here. So the blue bars are a 30-minute hive assay. So I've mixed the target and the beads together for 30 minutes. And unlike the red example, I get a very nice, clear distribution and difference between the signals. And the ability to resolve these in such short time periods for the assay comes from the fact that you can tune the amount of DNA on the capture probe surface. So in the red and the green assays, we've physically put and tried to block 100% of the binding capacity on the particle surface. Okay, I've put as much DNA as I can on the bead surface, and you can see the red and green results here. Now, if I change that to, let's say about 50% of the binding capacity, okay, so I haven't tried to completely fill all the binding sites on the bead, and I do that same experiment for a 30-minute hive, you get back to these blue data blanks. So tuning the capture probe density, tuning the assay time, and being able to resolve those small differences in the structures is quite a nice application and ability to do. Now, I put this in, do you need the number? Do you really care what the Zeta potential value is? It depends on what you're characterizing and what you want to get out. If you're just characterizing material, if you're interested in the surface chemistry, you want to know if the surface chemistry has worked or you've done a modification, then you absolutely want to do the number. If you're doing assays and you're looking at changes and relative changes, the question is, do you really want to have to go through the full process of giving the Zeta value? And this is a nice piece of work from Jeff's group, where they're doing small molecule detection on DNA modified beads, where they don't give a Zeta potential value, but the concept is exactly the same. We've just been talking about measuring duration. So full width half maximum here on the right hand side is an indication of the speed at which the particle goes through. You can see how they start off with my little annotation tool. So they start off with the top here with the nanoparticles, then they add stage 1, they add DNA to that, and you can see that there is a change in the full width half maximum. And then when that DNA that binds to its target, you see changes in both the size and the full width half maximum. And you can detect it, you can quantify the target in solution. You don't necessarily need to go all the way through to generate a Zeta potential value from it. So it's worth thinking about what you want and what does that number really mean to you as you go through this. Now, I put this in because this is, I'm sure this is I'm not the only one with a bugbear about this. And I think it's important that this is a growing community where there's far more users of TRPS than there was a few years ago when we started to do these kind of measurements. And knowing what people are doing and context is really important, because I bet we're not the only kind of people that get questions like this. Now, I'll give you a comment of a reviewer that came back to us, and I'm not going to say if this reviewer was right or wrong, because I think there are very good reasons why you would want to do what this reviewer is asking us, but I do want us to ponder and think about that, and just to reach out and make sure that we're all aware that there is a growing community of users. So you submit a paper based upon Zeta potential measurements, and the reviewer comes back and they say the paper deals with an important problem, and you think, fantastic, I've got this one through. And then you read to the next line, which is, however, an important aspect that is missing, and you think, oh. And is the comparison of the data obtained by TRPS with a reference method, i.e. go back and stick your sample in somebody else's machine and make a measurement. And I think there are good reasons why you'd want to do that in certain situations, and there are also equally good situations where actually this is probably a reviewer that hasn't come across TRPS, and as a community, if we educate more people, then it will make it far easier for us to start to publish. And as I say that, I say this because we've come across some really good examples where, and I think the name of this paper sums it all up, which is a systematic comparison, different techniques to determine Zeta potential in silicon nanoparticles. So people are doing the comparisons. There's lots of studies by many groups where we've made measurements on TRPS with Zeta potentials. We've put them in alternative technologies and made the measurements, and they can match. In some cases, if they don't match, is that because we have a different or a better way of measuring it? That's for us to discuss, but there is certainly enough comparisons out there in the literature now, where if people are simply asking you to validate this as a technique, there should be enough examples to offer a strong rebuttal to reviewers to say, actually, the community out there has tested this, and I would point you to some very good examples. This one being a good example of it, where you can illustrate to people where there are some good comparisons of doing this. So I want to end on some issues and problems and things that you might be aware of or should be aware of if you're making these and you want it to be reproducible. So we've got this electrophoretic speed of particles as they go through a pore. We know that these charged particles have a double layer structure, and the pore itself is charged, and it too has a double layer structure as it goes through. And any speed of the particles that goes through the pore is a function of the particle's zeta potential, but is also a function of the pore's zeta potential. So if you're doing an experiment, a complex reaction, and you've gone and modified the pore in some way, and it may throw off your results. You just need to be mindful. And I show an example of what a piece of the work that we've just had accepted, where we wanted to check, and this is, if you like, a good practice, an example that I thought I would share. We wanted to check that we hadn't messed up the pore, if you like. We hadn't modified the pore potential in some way. So this was an example where we've been doing analysing particles in quite protein-rich solutions. We've done this in 10% serum, 10% plasma. We've done 10 mg per mil protein concentrations. And I'll show you a couple of the examples of the proteins there that we've been using and how you can change the temperature of those particles in the solutions, and you can see changes in zeta potential. So this protein coronal structure is changing. But what I'm trying to draw your attention to is these green lines here are zeta potential measurements made on calibration beads after we've done the protein-rich sample analysis, after we've done the temperature experiment. So we calibrate the pore, we do a whole range of experiments, and then we run some other particles, i.e. our calibration particles, again at the end, and just pretend they're a sample. And we measure the zeta. So I get a calibrated pore and I measure the zeta potential of my particles at the end, and I show that actually those zeta potential measurements that we get haven't changed. The pore itself hasn't changed. It's not modified and changed the speed of the particles that it goes through. And the significance is, is this change between the red and blue bars because the pore's changed? Is it because you've applied a pressure? Is it because of something else? Or is it genuinely because the protein corona around these particles is different? Well, if I do all the calibrations and then I do the control experiments at the end, then that difference is definitely related to the experiment you've been carried out. So I think as a good practice, it's worth just running a particle through at the end. You can change the pore, and we've just submitted something for a Faraday discussion, which you can now download and have access to. And if you're thinking of how can I check what the charge is, or how do I get an indication if I modified the pore in some way, a really nice example and simple way to do it is to look at some current rectification studies of it. So you wouldn't necessarily need to go through the surface chemistry modifications which we put through in the Faraday, but you can simply measure the flow of baseline current, your standard bog standard electrolyte or a diluted version of your electrolyte, and measure the current flow through the pore surface. And I think the length and the rationale and the explanation is a little longer to go through, but I'm happy in terms of questions to come back and address this, or even after the event and come back. And but I would point you to the paper. And actually, if you're curious if your pore has been changed during your experiment, if it doesn't have the charge or that it started off with, a simple current rectification study that looks at the baseline flow, the baseline current flow through a pore, so just your KCl solutions, will give you a nice indication of if something has changed along the way. And we're relatively new into this area, and I've planned out 3 excellent groups if people are interested in this, which is Susanna, Celia, Henry White, and Jeff, who's also done some work on this, to go in and have a read. And it's a really nice example of working through, can you change the pore? Has the pore changed? And there's a simple QC method as you're going through this. So what I hope to give you is an introduction of Zeta and a flying visit of some of the techniques and processes that people have used to measure Zeta potentials with TRPS. Some current examples of people making Zeta potential measurements on TRPS. And again, I will acknowledge that I have not been able to cover the entire field. So if I missed your brief and your favorite paper, I apologize. But some nice examples that I've seen, and I hope to have shown you a couple of papers there where people have done some really nice comparisons between TRPS and alternative methods out there, so you can see they have been rigor checked. And some issues where, you know, if you're deviating in a way, you're using complex solutions and you want to check if the pole's changed, the particle's changed, or what you've got is true. Think about doing some simple calibrations or control runs at the end, or measuring current rectification studies. I get to travel around a little bit and go to conferences and talk to you guys on webinars, and really this is all because people in my group are really doing the work. And I imagine you probably wanted to watch this, but they're probably hard at work now doing this, so I'll just acknowledge them, saying, you know, Emma, Laura and Matthew have contributed to the data that I've presented today via their work, and the rest of the group are thanked as well. And IZON Science, the guys here, I've got to thank him, and Rob Vogel, of course, a huge thanks to Rob, and I hope I've done his process and therefore some justice. And I thank you for joining us, and I'm happy to take some questions or answer them later via email, and you're welcome to drop me a message and get in touch.
Thank you very much, Mark, for this very insightful presentation. I've got the one for you from Luisa Abramescu, and I think it's related to the latest part of your presentation, and it's how the pore size, the choice of calibration particles affect the Zeta potential results.
So how does the pore size?
Yes, the pore size and the choice of calibration particles affect the results.
So okay, I'm mute mine, or how would you want to do this? Okay. There we go. So how does the pore size and particles, and you've seen the effects of applied pressure on the system and voltages. So I guess the pore size, how would it affect the Zeta potential? Optimizing the signal so that you can see a really nice pulse, first of all, would be the key thing. So if you're missing some of the pulses, or it's too small and suddenly it's starting to restrict the particles as it goes through, with impact on its velocity. If you can see a nice pulse and you've got a nice calibration, then the pore size in a calibrated system should be okay. Now I say that within a stretch. So if I have a small NP 100 or a 200 and I've changed the stretch, and as long as I change the stretch and I can see a nice pulse, that should be, that won't really affect and impact too much on your measurement. If you change the pore size by going to say, okay, let's go to an NP 4000 and let's see if I can measure a Zeta potential value there, then it becomes a little more complex, because as I showed you in one of the earlier iterations and examples of Zeta potential measurements, if you increase the fluid flow through the pore, the flow of the liquid can completely dominate the speed of the particle. And when you go to very large pores, NP 1000s to 4000s, the flow of liquid through the pore is probably going to dominate the flow, the velocity. And I think that would be a much more complicated situation. So I think the smaller the pore, as long as you can see a pore state, you'll be fine. The calibration bead, what I think most of these examples that people have been publishing on, is negative particles. So if you have a system where you have a positive surface chemistry and you want to look at positively charged particles, you're probably going to have to match the calibration charge. You want to have a nice positive, if you like, charged calibration particle, and you want your particles to have, your calibration particles to have a really nice tight distribution. So you don't want a very broad distribution of your of calibration particles. In the calibration method, you're going to sample over 100 particles and take the average, and that's going to be your calibrate. So if your average of 100 odd particles is quite broad and you have quite a wide distribution, then that will impact on, if you like, the accuracy of the measurements. So nice tight distribution. Small pore sizes would be best, and a calibration particle where the charge matches that you think you have. That is, if you have a negative particle, you want a negatively charged calibration.
Thank you very much, Mark. Thank you very much, everyone, for taking part today. That's the end of this webinar. If you have any questions, just send them to me, and we'll pass them on to Mark. Thank you very much. Bye-bye.